Hello friends, and welcome to Webronaq. Here is a question worth sitting with for a moment. Right now, somewhere in the world, a programmer is writing code to tell a computer what to do. And somewhere else, a biologist is writing DNA to tell a living cell what to do. Those two sentences used to describe completely different worlds. They do not anymore. In 2026, the tools of computer science and the tools of biology have converged in a field called synthetic biology, and it is reshaping medicine, materials, and energy from the ground up. If you know how to think in code, you already have the mental model for this. In this video, we are going to unpack exactly how that works, step by step. Let us start with what synthetic biology actually is. Synthetic biology is an engineering discipline. Its practitioners read DNA, write DNA, and redesign it to give organisms new, predictable functions. That word predictable is key. Natural evolution is random. Mutations arise by chance, and natural selection filters them over millions of years. Synthetic biology flips that entirely. Scientists sit down, design a specific behavior they want a cell to carry out, and then write the DNA sequence that makes it happen. Think of it the way the National Human Genome Research Institute describes it: redesigning organisms to have new abilities. That could mean engineering a bacterium to produce insulin, or programming a cell to seek out and destroy a tumor. The raw material is always DNA, the molecule that stores biological instructions. And the goal is always the same: a living system that does exactly what the engineer intended. Now, what does that actually look like in practice? Here is where this gets genuinely exciting for anyone with a CS background. DNA functions like a programming language, and the parallels are precise. A gene is a function. It encodes a specific protein, just as a function encodes a specific behavior. A promoter is a switch. It sits just upstream of a gene and controls whether that gene gets turned on or off, exactly like a conditional flag in software. And a genetic circuit is a logic gate. Researchers at the New England Biolabs have shown, for example, that you can wire two promoters together, so a gene only activates when both chemical signals are present. That is a biological AND gate, running inside a living cell. The same Boolean logic you use in code applies here, just written in adenine, thymine, guanine, and cytosine instead of ones and zeros. So how do engineers actually build and test one of these circuits? That is the design, build, test, learn loop, and it is coming up next. Alright, so how do engineers actually build and test one of these genetic circuits? They follow a four-stage loop that will look very familiar to any developer. Stage 1 is design. The engineer models the circuit on a computer, arranging promoters and genes the way a programmer arranges functions and conditionals. Stage 2 is build. A DNA synthesizer physically assembles the design sequence, letter by letter. Stage 3 is test. That synthesized DNA gets inserted into living cells, and scientists measure whether the cells behave as predicted. Stage 4 is learn. The results feed back into the model, refining it for the next round. Design, build, test, learn. Sound familiar? It is essentially plan, code, run, debug, just running inside biology instead of a processor. And here is where it gets remarkable. AI is now automating this loop in ways that were unimaginable even a year ago. Now look at this. Because January 2026 changed the game entirely. A team at Rice University, led by bioengineering professor Caleb Bashor, published a paper in Nature, introducing a technique called CLASSIC. The name stands for combining long and short-range sequencing to investigate genetic complexity. Here is what it does. Instead of designing one genetic circuit at a time and testing it, CLASSIC generates hundreds of thousands to millions of DNA designs all at once. Every design gets mapped to its measurable output in living cells, producing a dataset large enough for machine learning to find the real patterns. The model can then predict how untested designs will behave before anyone builds them. Bashor described it as the first demonstration that AI can be used to design genetic circuits in human cells. That is the design, build, test, learn loop running at a scale no human team could match by hand. And that capability is already landing in real applications. So where does all this land in the real world right now? Two examples from 2026 show the scope. In February, MIT professor James Collins launched a 3-year, $3 million project pairing generative AI with synthetic biology to engineer programmable antibacterials. The goal is to fight drug-resistant bacteria, which kill hundreds of thousands of people every year. The approach uses AI to design small proteins that disable specific bacterial functions, then delivers them using engineered microbes, far more precise than any conventional antibiotic. Then, on July 1st of this year, the Department of Energy highlighted work by researchers at CABBI, the Center for Advanced Bioenergy and Bioproducts Innovation, at the University of Illinois. Their team combined AI, synthetic biology, and a biofoundry, a lab where robots handle the physical work, to significantly improve two industrial enzymes in a fully automated design, build, test, learn loop. Medicine and energy, two enormous domains, both being reshaped by the same convergence. Which raises a bigger question: why does this matter for the future of an entire field? So here is the through line from everything we covered today. Biology is becoming a programmable substrate. DNA is the code, genetic circuits are the logic, and AI is acting as the compiler, generating and testing designs faster than any human team ever could. The classic technique from Rice University showed this in human cells. MIT and the CABBI Biofoundry showed it landing in medicine and energy. And the market is responding. The synthetic biology automation platform market stood at $196 million in 2025 and is projected to nearly triple to $549 million by 2034, growing at 16.2% per year. For anyone studying computer science, biology, or both, this convergence is not a distant future. It is happening right now, and the people who understand both sides of it will define what comes next. Thanks for watching. See you in the next one.